My name's Tom Fenton. I've just finished 4 years of postdoctoral research in Copenhagen. I'm now starting my independent career in the Centre for Inflammation Research at the University of Edinburgh. And today I want to talk to you about a method that I developed which will give researchers a new way to analyse the intestinal immune system. So we actually discovered this by accident. I was working a lot of late nights in the gas lab with my colleague Peter Jørgensen. We were working on our own projects trying to understand different aspects of the intestinal immune system. We discovered that we could actually peel different layers of the gut wall apart. And then we realised that by staining this with methylene blue, we could actually identify the isolated lymphoid follicles, which are tiny lymph nodes embedded in the gut wall. So the human intestine contains a huge array of microbiota, some of which are good for us, most of which in fact are beneficial, but some of which are bad. The intestinal immune system has to differentiate between the good ones and the bad ones, and this is done in part in the isolated lymphoid follicles. And using this new technique, we're now able to isolate these follicles and analyse them away from other compartments of the intestine. So this new method allows researchers to look at the intestinal immune system in a way that hasn't been possible before. So we can isolate different parts of the gut wall, and these include the isolated lymphoid follicles, which we think are really important for the immune system to decide which bacteria to respond against. So this new method will allow researchers to isolate these follicles and analyse them during health and during disease. So far, we used the method to analyse the normal function of ILF in non-IBD patients. We found that the immune systems of the small intestine and the colon are somewhat separate from one another, and the ILF are driving a colon-specific response. Now we want to take it forwards and look at patients that have Crohn's disease or ulcerative colitis, and we want to see how these isolated lymphoid follicles are changing during disease and how they might be driving the disease. So we don't actually know what causes Crohn's disease or ulcerative colitis. We know there's a strong genetic component, there's an environmental component, and we know the microbiota are important as well. And we know that at least for Crohn's disease, the isolated lymphoid follicles play an important role. That's where the inflammation starts. But the actual mechanisms driving the inflammation in Crohn's disease and also in ulcerative colitis are not known. And this research will hopefully help us understand how these diseases are being driven.
3 reasons why I think this research is important. Firstly, this technique allows us to investigate the very early stages of Crohn's disease to allow us to understand how inflammation starts in Crohn's disease. Secondly, it also gives us the opportunity to focus on the very key immune cells which we think are driving this inflammatory process. Thirdly, and in the longer term, this work may potentially lead to a new treatment approach which is aimed at stopping Crohn's disease from happening in the first place. To me, this is a very exciting line of research in Crohn's disease.